So today's video is about membrane transport. The first thing we need to look at is cellular membranes. They possess two key qualities. The first one is that they are indeed semi-permeable, and therefore they are only permeable to certain materials. They're also selective, which means that there are several membrane proteins that can regulate the passage of material across the membrane. Now, there are two methods of transport. We first have passive transport, which does not require ATP, which is basically energy to move materials. Due to this, it usually occurs along a concentration gradient, so from high concentration to low concentration. And there are three main types of passive transport. You've got simple diffusion, which moves small or lipophilic molecules such as O2, CO2, and others. And then you have facilitated diffusion, which moves slightly larger or charged particles across membranes such as ions. And then finally you have osmosis, which moves water molecules. And then the second one is active transport, and that requires ATP. And there are two main ways. The first one is direct active transport, as you see in this image. It's when ATP is hydrolyzed to transport material. And the second one is an indirect active transport, which allows certain molecules to couple with another molecule and move along an electrochemical gradient, and we call this co-transport. So our first method of transport is simple diffusion, which is the net movement of molecules from a region of high concentration to a region of low concentration. And this will go along a concentration gradient. It usually moves small or non-polar molecules such as O2, CO2, and even glycerol. So diffusion will continue to occur until an equilibrium has been reached. Now, there are three main factors that affect the rate of diffusion. First off, we have temperature. So temperature affects the kinetic energy, of particles in a solution. Therefore, a greater temperature will mean a greater kinetic energy, and that would allow particles to move more quickly. Next, we have molecular size. So larger particles, in comparison to smaller ones, are subjected to greater resistance, and therefore will move much slower. Lastly, we have the steepness of the gradient. So the rate of diffusion will be greater if there is a higher concentration gradient. Yep. So next we have osmosis. It is the net movement of water molecules across a semi-permeable membrane from a low solute concentration to a high solute concentration, of course, until an equilibrium has been reached. Now, we know that water is a universal solvent, which means it's very important and will associate with polar or charged molecules. And because solutes cannot cross the cell membrane unaided, water will move to equalize two solutions. So next we have facilitated diffusion. It is the passive movement of molecules across the cell membrane via the aid of a membrane protein, which is what we have here. It is utilized by molecules that are unable to freely cross through the phospholipid bilayer. Usually these are large and polar molecules or ions. And there are two distinct types of proteins that control facilitated diffusion. We've got channel proteins and carrier proteins. So carrier proteins are integral glycoproteins, and they undergo a conformational change when the material binds to the protein, and this allows them to translocate the materials across the membrane. So carrier proteins undergo a similar attachment specificity to enzyme-substrate interactions, wherein only a specific kind of molecule can bind to the carrier protein. It is a slow rate of exchange, of transport, because it has to continuously undergo a conformational change. Then we have channel proteins, which is this one right here. And they are integral lipoproteins and they also contain a pore, which we see here. They are ion selective, and some of them may be gated, which we'll come to later. And they only move molecules along a concentration gradient because none of these use ATP. They do not use ATP. And they have a much faster rate of transport because the ions are just moving across the channel, the pore. So here we have the last one, voltage-gated ion channels. So they're basically integral proteins with a hydrophilic inner pore, which is this area right here. And in this case, we're looking at potassium channels, so potassium ions may be transported through the channel. The channel is comprised of 4 transmembrane units, but the inner pore acts as a selective filter because it's the narrowest region, and it'll restrict alternate ions from passing through. Potassium channels are also voltage gated, which means depending on the transmembrane voltage, they will either be open or closed. And this is very important for neurons and synapses. Here we have active transport. So active transport uses energy to move molecules against the concentration gradient. So there are 2 ways in which energy is generated for active transport. One is the hydrolysis of ATP, as we know, and the other one, as I mentioned before, is co-transport. So here's the general process of how active transport works. So first off, we have a specific solute that will bind to the protein pump on one side of the membrane. And then the hydrolysis of ATP to ADP will cause a conformational change in the protein pump, and this allows the solute molecule to be translocated across the membrane against the gradient. And it is then released. This is done via carrier proteins, so carrier proteins can both do facilitated diffusion as well as active transport. So the main form of active transport that we need to know is the sodium-potassium pump. So I'm going to walk you through what happens, how the sodium-potassium pump works using this image, and then I'll provide the written notes. So the first step, as you can see, is that 3 sodium ions will bind to the intracellular sites on the sodium-potassium pump. And then after that, a phosphate group, as you see here, is transferred to the pump via the hydrolysis of ATP into ADP. This then causes a conformational change, so now the pump is opened on the other side of the membrane, and it releases the 3 Na plus ions on the other side of the membrane. After that, we see this conformational change exposes the pump to K plus ions. So two K plus ions will bind to the intracellular sites, and then this phosphate group will be released, and that will cause the pump to return to its original conformation and allowing the potassium to be translocated across the membrane. Okay, so as you see here, I've written down the 6 steps for the sodium-potassium pump. Feel free to pause the video to take these down or visit our website for our full notes on all the units. So now moving on to bulk transport. First off, we have endocytosis. Endocytosis is the process by which large substances enter the cell. So what occurs in endocytosis is an invagination of the plasma membrane, which envelops this extracellular material. So here you can see the extracellular material, and this is, of course, the plasma membrane. The material kind of pushes against the plasma membrane to form this depression in the plasma membrane. And then the plasma membrane envelops all the material, and the invagination is then sealed off like this to form a vesicle inside the cell. Now, there are two forms of endocytosis. First, we have phagocytosis, which is for solid substances. And then secondly, we have pinocytosis, which is for liquid substances. And this here is an example of an electron micrograph that you might get. So lastly, we have the process of exocytosis, which is where large substances exit the cell. So here's a diagram of what occurs. Basically, there are vesicles derived from the Golgi, which fuse with the plasma membrane, like over here, and this allows the contents to be then expelled from the cell. So the vesicular phospholipids, these ones, are then added to the bilayer to replace the vesicular phospholipids used during endocytosis.